So, good afternoon. We're 15 minutes late for the program, but I think we'll still manage to have to end at the time we need to do. Now we have a panel session about why we're still waiting, exploring the slow pace of drug development for Sjögren. And I would like to invite Mrs. Professor Devesel in the floor. Thank you. And we have, thank you very much. And maybe we can start with the pre-recorded video of Dr. Thorsten Olski, which is the Senior Scientific Officer at the European Medicines Agency, who will share with us, through a pre-recorded message, the regulatory perspective on how innovation can be encouraged while keeping patients safe. Could we have the video, please?
Thank you very much for the invitation and for the opportunity to speak to you at this year's Sjögren Europe Patients Conference. And it would have really been my pleasure to present to you in person, but because of scheduling conflicts, I had to resort to a recording instead. First, about myself, my name is Thorsten Olski. I'm a Senior Scientific Officer at the Office for Scientific Advice at the EMA, the European Medicines Agency. And today I have the pleasure to give you a high-level overview over the workings of the European regulators and, very importantly, how this work contributes to innovation, to better medicines, and, of course, eventually better patient care. My background is in medicine as a dermatologist and in health economics. We have limited time, but nevertheless, we will have the ambitious goal that by the end of this presentation, you should understand at least the main principles of the European drug regulatory system. The operations of the EMA's committees, the role of the Scientific Advisory Working Party within the EMA system. And so to start with, the EMA is basically the European equivalent to the US FDA. It is one of the decentralized agencies of the European Union. The EMA was founded in 1995 and was originally located in London. But with Brexit, the agency moved to Amsterdam, and it is basically the scientific secretariat which is responsible eventually for authorizing and supervising drugs in the EU, and it operates with around 900 staff members. However, the main scientific decisions are taken by specialized committees, the abbreviations of which you see here: CHMP, CVMP, COMP, HMPC, and so on. These committees are furthermore supported by a large number of specialized working parties, and in all these committees, experts from all over Europe convene, over 4,000 in total, and it actually represents the regulatory backbone of the EU. Now, this structure has grown over the years, and it's always accompanied by an EU directive or an EU regulation. So for every committee, there exists an underlying EU directive or an EU regulation. Counterintuitively, in EU law, the regulation is in fact a stronger tool, and it is directly applicable to all member states. A directive is weaker, as it has to be followed by consecutive acts. The central function is, of course, the assessment of benefit risk and the authorization of medicines. This is done by the Committee for Human Medicinal Products, the CHMP, but the CHMP is not alone. In reaction to specific public health needs, other committees have been established for advanced therapies, gene products, tissue-engineered products. The CAT was established, the Committee for Advanced Therapies. In order to address the unmet medical need in children, the PDCO, the Pediatric Committee, was established. Rare diseases and orphan medicinal products are being dealt with by the COMP, the Committee for Orphan Medicinal Products, and so on. The idea is to support the development of new drugs over the entire course of the product life cycle. It is important at this point that the needs of the patients are the actual key drivers for all of these elements. These committees do not serve themselves. They are ultimately aimed at facilitating the development of better medicines, also for rare conditions. And only when those medications are available, the mission can be considered accomplished. Now, how do the committees work? As in the European system in general, each committee has to have a representative from each member state. So you will find an identical or similar composition as this one on the picture for all committees, with each member state being represented by one member and normally by one alternate. Some committees do not have alternates. These members are nominated by each member state, be it by the health minister, the head of the national agency, or similar. The alternate only steps in when the member is not available, and voting does not take place proportionate to population of the member state, but instead each state has one vote. So from amongst their members, the committee elects a chair and a vice chair. Now, the EMA is supporting drug development over the entire course of the product lifecycle. And the Scientific Advice Working Party, abbreviated SAWP, is embedded in this process. Whereas the CHMP and its Pharmacovigilance Committee, the PRAC, take decisions at the time of evaluation of an application or in the post-authorization phase, the COMP, the CUT, the PDCO, and the SORB contribute during the earlier stages of the development. But of these, the scientific advice is the core instrument of the regulatory support during drug development. Particular interest has been put on the contribution of patients and healthcare professionals. In order to make sure that their voice is heard, the European Commission appoints a specific number of patients and healthcare professionals as full voting members to certain committees. In addition to this, the input from healthcare professionals and patients is sought product per product on an ad hoc basis. So as you can see, the input can be sought as early as at the CUT or at the SORB level, or it can be sought later at the time of marketing authorization at the CHMP. So you see how crucial and important it is for the agency to listen to the voices of patients and to take their views into account when discussing new developments. The EMA is making a strong effort to support research and innovation with an entire ecosystem of offerings, which are meant to facilitate the process for applicants and smoothen the way towards getting a marketing authorization. For the often neglected population of children, companies submit a pediatric investigation plan, a PIP, which defines how a drug is being studied in children. For advanced therapies, companies can be supported by the CUT. And very innovative medicines can take the route of the PRIME scheme, the Priority Medicines Program. Now, this will come with the advantage of getting an early nomination of a CHMP rapporteur, a dedicated EMA contact point, as well as the occasion for kickoff meetings and close interaction along the development route of a product. But companies can also request scientific advice for a specific drug, or can request a qualification of a novel methodology, which would also include novel endpoints. So a sort of scientific advice light is offered with the Innovation Task Force. Now, the Scientific Advice Working Party is a committee, which is an advantage, because its members do not have to be represented for each member state one, but instead the nomination can take place based solely on expertise, and drug developers can request scientific advice only based on their strategy. The scientific advice does not pre-assess actual data, and applicants will only receive a feedback on questions they have posed specifically. Now, there is an option of coming back for follow-up advice, which is supposed to open something like a continuous dialogue between regulators and drug developers. So our advice to drug developers is to come early and come often. Now, the EMA offers fees incentives for pediatrics, for orphan medicines, for applications filed by small and medium enterprises, and so on. Furthermore, there is the possibility of requesting parallel joint scientific consultations with HTA bodies, as well as parallel scientific advice with the US FDA. Now, the procedure takes between 40 and 70 days, and it ends with a final advice letter, which is being adopted by the CHMP. So it becomes an official document of the CHMP. The advice received, however, is not binding per se. It remains a recommendation to the applicant. But our own analysis have shown that following the recommendations of the Scientific Advice Working Party, it was a clear predictor of success of the subsequent application for marketing authorization. So in a way, the scientific advice has also been partially victim of its own success, as the entire process has not skipped the attention of the industry, and so we have seen an ever-increasing number of applications from industry over the years, as seen in the chart on the lower right. Now, we intend to maintain our timelines for applications, but this substantial workload has still to be absorbed by the European system, by all member states, so it's a bit of a challenge. But in order to provide the best advice possible, the SORB will not just discuss amongst themselves, but it will rely on input from other committees, the COMP, the PRAC, the CAT, etc., and will also consult working parties, the methodology working party, and so on. So the idea is, of course, to ensure regulatory consistency across many advices. But with this brief overview, I do hope that I was able to give you just a glimpse of the work of the EMA, and I hope especially that you've gotten an idea of how much motivation there is behind the system to address the medical needs of patients and how much emphasis there is on getting the views of patients and healthcare professionals during the early developmental phases. So with this, I thank you and I wish you a good meeting and a successful meeting. Thank you very much.
So now we're going to have Professor Valérie Devoucel, if I pronounce your name well.
Yes,
Who is Professor of Rheumatology at Brest University Hospital in France and head of the Reference Center for Rare Systemic Autoimmune and Autoinflammatory Diseases. And she's a leading clinician and researcher in Sjogren's and autoimmune diseases.
Thank you. So Joyce, I remember that for the first meeting, during the afternoon, you tell us to turn, to move a little bit, just to be better concentrate. So I hope you are not sleeping. And I'm going to do as Nathan, try to pass my slide. I think it's like that. Yes. So you ask me why so slow, just at the moment when everything happens. So here's where I am, located in France, in the western part of France. And of course, for years, we have a big organization, clinical and in the research department to work on Sjögren's disease. So that's the big group now, and we are happy to do that for years. So why so slow? Maybe because of patient heterogeneity. So you heard about the Zika symptoms, the systemic symptoms, and Geoffrey showed you just a minute ago that SSA positive, SSA negative could change the way of the patient and the trajectory. So we have this problem also in research, which kind of patient. Some are more considered as Zika, the other as systemic, and all the patients have fatigue, and Zika symptoms are the main symptoms of the patient. But in fact, if you are looking at the molecular characterization that can be done by Nathan, for example, you can see that there are a lot of ways to have a Sjögren's. So sometimes it's difficult for the clinical trials. And I don't speak here about damage and activity, and I can speak about pre-Sjögren, because you heard that we don't know exactly when it begins, so maybe we don't know exactly when we have to introduce a drug. So there are some research field actually concerning pre-Sjögren to catch the data before the beginning. That's not really easy. And don't forget also that some patients have real Sjögren. I don't know where disappear. Real Sjögren, but they don't fulfill the classification criteria, so they are not included in study. So why so slow? Also because we don't want to stop. We want to stop some different things in Sjögren. It could be the fatigue, it could be the systemic activity, but it also could be the activity in the tissue. And why so slow again? Because some domains of interest maybe are low, take time to improve. Quality of life, probably, and difficulty to sleep or to improve salivary flow. And why so slow? Also because we use sometimes some old-fashioned way to evaluate the symptoms. We still evaluate Zika dryness with this test. However, we have some more modern tests to evaluate it. So maybe in the future, we could use this. It is more precise to know how many drops you have in your eyes, for example. And we also can imagine using other way to collect, to collect the symptom of the patient with some specific tools, for example, the PEPS tool. So finally, we previously write some recommendation to how to treat patients with what we had before. And we write some international recommendations, some French recommendations. I suppose there are some Greek recommendations, etc., according to the treatment we can have in each country. So with this kind of recommendation, we explain how to treat different types of symptoms. And I just show you this because, of course, we speak about treatment of dryness of the eyes, of the mouth, but also from this kind of dryness. And Coralie was involved in the PNDS in France for the recommendation, and she was acting as a patient representative. And she told us, please don't forget this. So, for example, you did something. We had a lot of recommendations on that. And this is very important. And of course, we explain how to deal with some other involvement, and we add some specific data for the patient, how to do by yourself to have less parotidomegaly, for example. So now, can we change the course of the disease? Because before, we couldn't. It was not possible. And you can see here the previous trial. We did a lot. Alone, I want to say without the company for most of them. We did this trial, and most of them failed or don't give enough results. And now we have a lot of new treatment evaluation with a lot of positive results. So we are not so slow. Now it's increase and increase and increase. I'm going to show you some of the results. So I'm not going to detail all that, but for the patient, you have to know that we have, or the company, developed a lot of different drugs, and that results are now positive for many of them. So the process is long between good results and medication that we can use, but there are a lot of drugs that gave us good results. So, for example, the ianalumab, it's a treatment against B cells because you understand that the B cells are activated, they produce autoantibody, probably they participate to the destruction of the gland and the risk of lymphoma. So it is a good target. And with this treatment, a monoclonal antibody that blocks, I cannot move. I don't like to stay like that. So that blocks B cells, and also the way using by B cells to stay alive for a long time. Yes, I don't have. The B cells are really depleted in the blood. And you understood also that there are different phases for a treatment. So the phase 1, to be sure it's safe. Then the phase 2, it's a larger study with large number of patients and different dose evaluation. So the phase B for this treatment was positive. This is here the systemic manifestation. This is here the this is here the salivary flow. So it increased, and it was maybe the first time that something like that appears, so it's very, very important. And this is the PRO on the table, so the symptoms feeling by the patient. It does not really increase, but they have more saliva flow. So after the phase 2, you have the phase 3 before you can use a treatment. And the phase 3 are finished now. They are dedicated to Zika symptoms or systemic symptoms. We don't have yet all the results, but the firm communicates and told us that the results for both studies are positive. So that's really a good news. And you will see the results, the complete results, at the American Congress in 1 month. And also you will see some results about what's happened in the gland, because you understood that it's important for the doctor to understand what's happened. And you will see also some results about what's happened in the gland and in the blood. Another target is CD40 and CD40 ligand. You can see here what's happened. In fact, 2 cells, an immune cells, B or T cells, interact with another cells to activate immunity. And in Sjögren, it is too more activated. We want to block that. And this is a way to block this interaction. It was a surprise because we were not thinking about this ligand, but it has a sense because it blocks interaction between cells. And the results also with such a treatment was positive. So the phase 2, you remember, a larger number of patients and a dose evaluation. And the result was positive for the systemic manifestation, so the SDI. You can see here the SDI. It was also positive for the ESSPRI, so the symptoms of the patient. And it was also positive here on the, the left side for the salivary flu. So again, some very interesting results. And we have a small idea of what's happened in the gland because it modifies some marker that favor the development of lymphoma. But we don't really understand exactly what's happened. We have just 1 or 2 data on it. Another firm also developed such a treatment with similar results. So also that's good. That means that results are really solid. Another way to block B cells, T cells, and autoantibody is to use this type of treatment. This is an IgG. It's something that we all have in our body, an immunoglobulin, so like an antibody, we have it, and they die in the cells. But there are a specific mechanism that catch the antibody, the immunoglobulin, it catch it, and it put it again in the blood. So it's to protect us. For example, if I have some antibody against tetanus, I want to keep it. So it's a way to keep it long time ago, okay? So in Sjögren, we don't want to have autoantibody for a long time because we think they induce the disease. So with this treatment, we blocked this survival mechanism. Okay, so autoantibodies don't stay alive a long time in the blood. So this is a very interesting way to interact with Sjögren disease. And again, a phase 2 last year was positive, and the phase 3 is ongoing in lots of countries. Some other similar treatments are evaluated also in Sjögren. And it's nearly the last slide, not the last, but nearly the last one. And you can also act into the cell, okay? So antibodies, B cells, okay, we try to block them, but you can also act into the cells. So they are alive, but you block the system inside the cells. So it is, for example, the BTK or the TYK2. It's something inside the cells that activated the cells. So we have some preliminary results with such a treatment. With good results, you can see SDI, oh, it's in French, sorry, SDI, salivary flu, and symptoms that were not improved. This is another way to act into the cells. And the cortisol, so I'm not going to develop it because it has been done previously, but it's also a very interesting way to block B cells, and for a long time. The procedure is difficult, yes, but it acts for a long time. We think that maybe it can be a sort of reset. You all have reset on your computer, so it could be that. And the side effects are lower than in cancer because you are not going to destroy a lot of cells. Just a low number of cells, but pathological B cells. It's the end, Cathy. I'm afraid of Cathy. So, I hope you agree that Sjögren is probably no more an orphan disease. So I'm going to sleep now. But maybe not really. So we can improve the tool to assess the disease, of course. When first treatment will appear for the patient, we can design other trials, for example, to evaluate other symptoms, to be sure that it has efficacy, I don't know, on dryness, of SSA negative or vaginal fatigue, or etc. So when you have the drug, it's easier after to design other trial for secondary endpoint. And of course, experts are going to work on stratification. Huh? You understand we like stratification. And of course, now education and patients association, again and again, to explain how to deal with the drug. And for the ongoing, for the future, unfortunately, I'm not going to retire just now. But what's happened in the gland, in the B cells? Are we just going to stop the symptoms or to stop the abnormal B cell for a long time? Is it clear that we are going to avoid lymphoma or not? And we still know that, but we know that now that we can restore partially the salivary gland because the patient improves their salivary flow. So maybe we can restore the parenchyma of the gland. So thank you very much.
Thank you very much, Professor. Do we have any question? I have 3. But I'm going to do one only. Okay, and can you tell us what do you see as the main reason drug development for Sjögren's remains so slow? What did you see as the main reason? What is the main reason of so slow development of drug?
What was the main results? Because it's simple. It's because Sjögren is very heterogeneous. It's difficult to know which endpoint you are going to use in a clinical trial. Is it Zika symptoms? Is it lymphoma? Is it arthritis? So it was difficult, like in lupus or something like that. So that's why it took time.
And how can patients and their organizations better support the research process? How can patients and
doing this, yes. Being there, to explain to the patient what will happen, being also together. That's important because it's not the end of the research. I know. Yeah.
Okay, and the last and tricky questions.
Yeah.
What gives you hope that we are getting closer to real progress? The clinical trials phase 3?
Oh, yes, of course. Yes, of course. I think the phase 3 is the best way to obtain the treatment. So yes, we are all happy about that.
I was just wondering, you know, there are a lot of benefits for orphan diseases in drug development. Are you concerned about shifting the perception away from it being an orphan disease and, you know, considering that there are potentially different subtypes and, you know, all the complexities? Just curious.
It was my first topic on the first Sjögren meeting. So there is some discussion about is it a rare disease or is it not a rare disease. In Europe, it is considered as a rare disease or an orphan disease. And in some other country, it is more frequent, maybe. But we have, you're right, in the concept of the rare disease, some patient with different subtypes. So probably, I don't know exactly the percentage. I don't know if SSA negative are more frequent than SSA positive, but probably, yes, it's equal. Half. But there are subtypes here, clearly, yeah.
One remark, Valérie, thank you for your kind words about patient partnership. I take this opportunity to tell you again and again, and that has been said by Professor Goules this morning in the workshop on clinical trial. We are working closely with Valérie's team, with Professor Andreas Goules' team, with the UK teams, with every team in Europe. And we saw, and Professor Goules told it this morning, that there's a real switch in designing the clinical trial. And when I see all the negative trials from 10 to 12 years ago, as Valérie showed earlier, I wonder, Valérie, if you run the same trials today with, let's say, the new partnership you have with patients, with the new discoveries of finding, do you believe that the rituximab trial, by example, will be shown as negative if you include another arm, by example, only for the symptom burden and not only for SDI damage and things like that? What do you think? Do you think that if we take the previous trial, we do it again today with what you know, with how we collaborate with the team, do you think it would be still negative? Tricky question again. Sorry.
I think the best way, I don't know for rituximab, really I don't know.
It was just an example.
Yes. But rituximab gave us confidence on B cells target because there were some endpoints that were positive. And now we have some more powerful drug acting on B cells. So I think we have to focus on this. And yes, working all together to design some endpoint was an improvement, of course.
So let's continue.
Okay. Thank you very much, Professor, for your dedication to Sjögren's, to our community. Yeah. Thank you very much. And now it's the official end of the first day. We managed to have it on time. Please be known that all those who have registered for our sightseeing and dinner, they have to be at 4 o'clock at the lobby. 4 o'clock exactly, the Pullman, will depart, okay? 4 o'clock at the lobby. Thank you very much.